I'm going to do a quick demonstration to try to help you understand why it is that one individual can produce so many genetically distinct kinds of gametes. So instead of using chromosomes, I'm going to use playing cards. And I'm only going to use 8 cards because if I tried to emulate the example in humans with 23 pairs of chromosomes, 46 cards, it would just be unwieldy. So I'm going to pretend that there's an organism called the CardShark that has 4 homologous pairs of chromosomes. So we can think of these cards or chromosomes as being either from the mom if they're this pink color or from the dad if they're this blue color. And a homologous pair of chromosomes, for instance these 2 aces, would have the same genes in the same locations but possibly different alleles. So if there's a gene right here for fin length, there's also a gene right here for fin length. This allele might be for short fins, and this allele might be for long fins. So a homologous pair of chromosomes means one from mom, one from dad. They carry the same genes, but possibly different alleles. So if we were to look at a somatic or body cell in a CardShark, we would see that there were 8 chromosomes. But when that CardShark undergoes meiosis in its little CardShark gonads to produce gametes, you'll end up with 4 chromosomes. But it's not going to be completely random which of those 4 chromosomes go into the gamete. So it's not like you just take your 8 cards, mix them up, and pull out 4. Instead, each gamete will have 1 member of each homologous pair. So in order to be happy and healthy, the CardShark needs ace, deuce, 3, 4. But when it does that, it's completely random whether it puts into the gamete the chromosome that the CardShark itself got from its mom or its dad. So the origin of the chromosome being maternal or paternal is completely random. And that happens at metaphase 1 of meiosis. And this is a source of a lot of genetic diversity, this random alignment of homologs at metaphase 1 of meiosis. So if we try to show metaphase 1 of meiosis by lining our cards up as homologous pairs, when I see a pair I just stick them together, and we think of this as being the metaphase plate in a dividing cell. This is one pole, this is the other pole of that cell. These homologs have aligned in pairs across the equator. And now if we flip them over to see whether they're from the CardShark's dad or mom, we can see that in this example, oh my goodness, the CardShark will produce a gamete after meiosis 1 finishes, and these chromosomes go through meiosis 2 to produce a gamete. That gamete will have only chromosomes from the CardShark's dad. Now, if we mix up the cards again and assume that the CardShark undergoes meiosis again a little bit later that afternoon or whenever, and we align these up again at metaphase 1 of meiosis along the equator, and we flip these cards over again, this time we'd produce a gamete that has two chromosomes from the dad and 2 chromosomes from the mom. So a CardShark with its 4 chromosomes can actually produce 2 to the 4th or 16 different kinds of gametes relative to maternal and paternally inherited chromosomes. Now humans with their 23 pairs of chromosomes can actually produce 2 to the 23rd or over 8 million different kinds of gametes. So I hope this helps you understand that random alignment of the homologs at metaphase 1 of meiosis is a source of great genetic diversity in terms of the kinds of gametes an individual organism can make.